Before I start my presentation, I would like to thank Jenna and Pasquale for organizing this wonderful symposium and inviting me to speak today. In my presentation, I will explore how brain regions, neurons, and their networks that support cognition develop, and how they have evolved along the human lineage. Many studies have shown that cognition emerges from neural activity in large-scale association networks of the cerebral cortex. These networks integrate sensory inputs and shape voluntary behavior through outputs to motor system. So it is not surprising that most of the human cortex is association cortex, as shown here in light magenta, while green, primary sensory areas, and red, primary motor area, are comparatively small. They are like islands in an ocean of association cortex. In other words, most of our cortex is dedicated to cognition. Information typically flows from primary sensory areas to higher order association areas in a hierarchical pattern, along what is often described as a sensory-to-association axis, as illustrated here for the visual system. Neighboring areas exchange feed-forward and feedback signals, enabling progressively more complex feature extraction. And in many ways, it is intuitive how these hierarchical sensory networks are constructed during development by experience and activity-dependent input from the peripheral sensory organs. However, this tidy hierarchy becomes less apparent in higher order and transmodal association areas, which connect over long distances to the prefrontal cortex and other association areas. These connections are not hierarchically organized, but rather distributed, with many axonal pathways crossing the entire cortex and bypassing lower order and primary sensory motor areas to converge on the prefrontal cortex and other higher order association regions. The prefrontal cortex acts as an integrator. It receives input from diverse regions, selectively gates and combines that information and sends feedback back to many of the same regions. This top-down feedback helps regulate attention, thought, emotion, and actions. One reason that prefrontal cortex is often described as the CEO of the brain. The dominance of the prefrontal cortex and broader distributed association networks in human cognition can be traced to three major evolutionary adaptations. First, only humans, other apes and monkeys possess a true prefrontal cortex occupying the lateral portion of the frontal cortex shown here in blue. In all other species, regardless of how large or small is their cortex, the most anterior part of the lateral part of the frontal cortex is instead occupied by motor areas shown here in red that drive voluntary movement. Second, humans have the largest prefrontal cortex and association cortex among living primates, both in absolute and relative size and terms, as shown in these plots. Third, pyramidal neurons, which are the principal cells of the cerebral cortex, have more dendritic spines in the prefrontal cortex, reflecting a greater number of synaptic connections than pyramidal neurons elsewhere in the cortex. Importantly, this anterior to posterior gradient is substantially steeper or exaggerated in humans compared to all other analyzed primates. Taken together, these adaptations mean that humans have an unusually large and highly connected prefrontal cortex that exerts strong influence over temporal and parietal association networks, supporting cognitive capacities unmatched in other animals. And in the rest of my presentation, I will summarize our work on how the prefrontal cortex and broader association networks are built through mechanisms and principles distinct from those shaping sensory motor networks. I will also show how competition between these mechanisms shapes the development and evolution of cortical sensorimotor-to-association axis. Our journey started almost 20 years ago when my colleague Matt Johnson, now at the Broad Institute, and Yuka Imamura Kawasawa, now at the Penn State University, used microarrays to profile gene expression in tissue samples of primary sensory motor and association areas shown here in green and magenta, respectively, across the full course of human brain development. Using this approach, they discovered that emerging primary sensory motor and association areas already exhibit distinct transcriptional or gene expression signature by early to mid-fetal development. Importantly, these gene expression differences were present while most of cortical neurons were still being generated and before extensive synaptic formation had occurred. What they found revealed two major and previously unrecognized patterns of gene expression. First, the two broad gene expression gradient that emerged from frontal and temporal poles and basically preferentially marking future association cortex. And shown here are two examples of these gradient genes. Cerebellin 2, which is a highly enriched in future prefrontal cortex, and it's known to be a synaptic organizer, and MPY, a neuropeptide to be enriched in temporal-occipital part of the cerebral cortex. Second, more focal region-specific gene expression was also observed in prospective sensory and motor areas. And overall, a few hundred genes were identified and grouped into modules based on similarities in their spatial and temporal expression dynamics across emerging association and primary sensory motor areas, as shown on the right. Based on these opposing gene expression patterns, we hypothesized that primary sensory motor and association areas are shaped by distinct, specialized developmental mechanisms. Looking more closely at these gene co-expression groups or modules, we notice that their spatial-temporal patterns shift dramatically as cortex expands or grows. First, the two association gene modules emerge from the frontal and temporal poles and initially shared gene expression signatures with neighboring allocortical regions such as hippocampus and amygdala. Secondly, we found that these programs appear to move inward with age as development proceeds, and they start near this pericentral region neighboring allocortex, and as the cortex grows, shift toward the geometric center of the expanding cortical shield. But what was surprising is that they consistently avoided the parts of the cortex or the region that will become the primary sensory and motor areas. And because they actually emerge from these peripheral regions of the cortex or pericentral with allocortex, we name them pericentral identity programs. In contrast, the centrally positioned region that will give rise to primary somatosensory areas such as visual, auditory, somatosensory, and motor exhibited a very focal and distinct set of genes whose expression remained stable over development, and we call these central programs. Our hypothesis is that these opposing programs held pattern cortical areas and networks to establish the broader sensorimotor-to-association axis of the cerebral cortex. And consistent with this hypothesis, we found that even a relatively small set of few hundred pericentral association and central future sensory motor genes is sufficient to reveal striking developmental shift in topological relationship between cortical areas across human development. Early on, these expression patterns suggest that the cortical areas are being topographically organized primarily along frontotemporal axis. Over the time, this axis is gradually transformed into the familiar hierarchical topology along the sensory motor to association axis, which ultimately becomes the dominant functional feature of the adult cortex. Together, these findings suggest that these genes are not only essential, but may also point to previously unrecognized mechanisms for building cortical areas and their networks that connect them. So, consistent with this hypothesis, we found that genes involved in retinoic acid signaling, a pathway not previously implicated in cortical areal or circuit development, are enriched among genes specific to the frontal, pericentral, or association gradient or gene module, shown here. We observed that many genes related to retinoic acid signaling, including its canonical nuclear receptor, RXR and RXRG, are basically highly enriched in the fetal prefrontal cortex, but not in the motor cortex. And in contrast, we observed that a gene called CYP26B1, which encodes an enzyme that degrades retinoic acid, is enriched in future motor cortex. Based on this finding, we hypothesized that retinoic acid signal is strong in the fetal prefrontal cortex, or which is the association part of the frontal cortex, where it regulates the expression of key developmental gene, and that this signaling is spatially restricted to the developing prefrontal cortex by RNA degradation in the motor cortex. In this model, CYP26B1 mediated retinoic acid degradation, shown here in red, limits the spread of prefrontal area identity into adjacent cortical areas or territories that are normally specified as motor cortex. To test this hypothesis, we first showed that retinoic acid signaling is present in the developing prefrontal cortex. Because retinoic acid is known to regulate gene expression through its nuclear receptor RAR and RXR, we perform cut and tag assay to identify genes that are directly regulated by these receptors. And in addition, we also analyze differential gene expression in mice that lack two key retinoic acid receptors, RAR beta and RXR G, which we previously found to be enriched in developmental human and mouse prefrontal cortex have shown that are essential for proper development of the mouse prefrontal cortex. So together, these approaches reveal that retinoic acid signaling controls a set of genes called gene regulatory network, and that are enriched in the prefrontal cortex. And among these genes that are directly regulated, many of them have been shown to encode axon kinase molecules and synaptic molecules, as well as disease risk genes associated with autism and schizophrenia. Among targets of retinoic acid signaling in prefrontal cortex, we were able to demonstrate experimentally that cerebellin 2, which regulates synaptic formation specifically in the prefrontal cortex, and we were able to show that it actually does that through a hominin-specific gene regulatory mechanism. What I will in the next slide talk is about another gene that I will highlight, which is called MACE2, and which is a key developmental transcription factor selectively expressed in prefrontal cortex, frontal pericentral association programs, as well as previously linked by other groups to autism and intellectual disability. So consistent with a regulation by retinoic acid in the developing human prefrontal cortex and the mouse, we found that MACE2 is selectively enriched also in a developing mouse prefrontal cortex, shown here on coronal. You can see that expression is present in the medial prefrontal cortex, a little bit orbital, but it's actually largely missing from what should be a motor cortex. And also we found that this expression is dramatically reduced in mice lacking the two key retinoic acid receptors, RAR beta and RXRG, which we previously shown are essential for proper development of mouse prefrontal cortex. And so to investigate the effects of MACE2 loss on prefrontal cortex development, we generated a conditional knockout mouse in which MACE2 was selectively deleted in cortical neurons. As highlighted in these whole mount in situ hybridization images, which highlight restored gene expression patterns, mice lacking MACE2 show a loss of prefrontal expression of cerebellin 2. You can see that a key prefrontal markers that we have also shown is under the control of retinoic acid signaling and is essential for making synapses in the prefrontal cortex and connectivity. In addition, expression of CYP26B1, which was expressed a little bit more posteriorly in premotor and anterior insula, this expression was shifted medially into a region where normally prefrontal cortex would be located in mice. And consistent with these molecular changes, we observed that the corticospinal tract, a major pathway that controls voluntary motor behaviors, and that normally originate exclusively from motor and premotor cortex, now ectopically originates from the medial frontal cortex, where normally you would see the prefrontal cortex. This is striking because the prefrontal cortex does not typically give rise to the corticospinal tract in adult mice. So together, these shifts in the expression of areal markers, as well as the dramatic changes in the connectivity of these mice, and several other features that I did not have a chance to show you, indicate that the motor and premotor territories expand frontally along the motor to prefrontal association axis, effectively replacing the prefrontal cortex. In other words, maze-2 mice lack a typical prefrontal cortex, further supporting a model in which retinoic acid signaling is essential for proper development of prefrontal association cortex. Our finding indicates that retinoic acid forms a signaling gradient in the developing prefrontal cortex, and that selective expression of its degrading enzyme CYP26B1, shown here in red, in the human premotor and motor cortex restricts posterior expansion of this gradient. To test this experimentally, we first ask whether mice show a similar pattern by examining CYP26B1 expression in developing cortex. And consistent with the typical arrangement in which the prefrontal cortex occupies only the medial and orbital part of the mouse prefrontal cortex, and motor cortex occupies the anterior or the lateral part of the frontal region, CYP26B1 expression in mouse frontal cortex was shifted anteriorly and displayed lateral to medial gradient, avoiding what would become the medial prefrontal cortex. And so we hypothesize that this is also prevented, and to test this, whether CYP26B1 limits the spread of retinoic acid signaling into lateral frontal cortex, we generated mice lacking CYP26B1. Using a rare LAG-Z reporter that registers retinoic acid signaling in vivo, we found that CYP26B1 knockout mice show increased retinoic acid signaling with a gradient that expands dramatically from medial prefrontal cortex, which you would normally find retinoic acid, into lateral frontal territories that normally would be occupied by premotor and motor areas. Consistent with this lateral expansion of retinoic acid signaling, we observe broadened and elevated expression of cerebellin-2 and maze-2, remember the two targets of retinoic acid in the prefrontal cortex, as well as the precocious and lateral expanded projection from the medial association thalamic nuclei in other territories that are occupied by this expanded gradient. And so together these results show that the patterning along the motor to prefrontal association axis is regulated by competition between enriched retinoic signaling in the future prefrontal association cortex, and CYP26B1 mediated degradation, which exclude retinoic acid signaling from the emerging motor cortex and thereby constraining prefrontal identity. Because retinoic acid signaling and key prefrontal genes are largely excluded from developing motor cortex, and opposing mechanisms can shift the identity between motor, which is an association cortex, we hypothesize that the type of competition exclusion may more broadly pattern primary sensory and other association cortices during development. And so to test this hypothesis, we return to our list of genes expressed in either pericentral association or central sensory motor programs in the developing human brain, and we focus on genes near the top of this list that encode receptor or ligands with anti-correlated expression between association and sensory motor cortex. This analysis identifies several ligands pairs, including semaphorin 7A and plexin C1. These genes function as axon guidance molecules with mutually repulsive effects, and we found that SEMA7A was enriched in primary sensory and lower order areas, while the plexin showed an anti-correlated expression enriched in higher order association areas and allocortex, as we predicted based on their expression. So we next hypothesized that this selective enrichment of semaphorin in primary areas is induced by thalamocortical axon from first order sensory nuclei, which relay direct input from peripheral sensory organ. And to test this, we actually we basically screen almost 100 knockout mice to find mice that do not have a input to from thalamus to the cortex. And we have identified such two mice, mice that lack SATB2 and ZBT18 transcription factor. It is not that important why they lack, but it's important they lack these projections. And so strikingly, consistent with our hypothesis, these mice lack primary sensory area, shown here by total lack of SEMA7A, as well as other markers of primary area. Instead of the cortex is broadly occupied by plexin C1 and other key genes that are associated with the association programs. And projection to prefrontal cortex now become widespread in the manner of reminiscence of distributed association networks. So these results support a model in which pericentral association programs emerge from the frontal and temporal pole, share molecular properties with neighboring allocortex without requiring thalamic input or induction. In the contrast, the central sensorimotor programs are induced by thalamic input that claim primary cortical areas. And together, these opposing mechanisms then interact to shape the development and evolution of cortical sensorimotor axis. We call this model MIND, stands for multinodal induction and exclusion network development. Multinodal reflects the presence of multiple primary area nodes and the shared organizational principles of exclusion, induction, and exclusion between sensorimotor and association. So I will close my presentation by summarizing how the mechanism principle encapsulated by this MIND model provide a simple explanation for this proportional expansion of the prefrontal cortex and broader, largely transmodal association networks. According to the MIND model, the early developing cortex is patterned by two major gradients of wave of pericentral association gene expressions that propagate from frontal and temporal poles. And these gradients polarize the early cortex and organize distributed association networks along the dominant frontal and temporal topographic axis. As the cortex grows and the association gradients expand and occupy territory that would not be specified or occupied by central sensorimotor areas shown here in black. And once established, basically we create an ocean of association cortex and islands which are excluded by exclude association program. And although the human cortex is roughly 3 times larger than that of the chimpanzee, our closest living relative, this expansion was not linear or isometric. Primary, actually, sensorimotor cortices did not enlarge equally. And basically, the MIND model offers a simple explanation. During human evolution, cortical and brain size increased, as that's well established, while body size and many peripheral sensory structure became smaller or remained relatively stable or scaled with the negative allometry related to the brain and the cortex. And consequently, less cortical territory was required for primary sensory motor representation, leaving more territory available for pericentral association programs to occupy and form the distributed, largely transmodal association networks. I don't want to leave you with the impression that allometric scaling is the only key to human evolutionary success. Our group and others have also profiled changes in cell types and their gene expression using single-cell assays across many regions of the cortex, including dorsolateral prefrontal cortex work from our lab. And so collectively, these studies show that all major classes of cortical neurons and glial cells are conserved across humans, chimpanzees, and even macaques. And at the same time, nearly all of these cell classes exhibit broad and in some cases cell type specific, human specific shifts in gene expression. And you can see on the right a few examples of human specific changes in cortical neurons that our group has identified. And these expression changes have been linked to and are predicted to alter molecular programs, electrophysiological properties, and signaling features of human neurons, as well as their synaptic connectivity. For example, evidence consistent with our finding that dopamine neurotransmitter is being synthesized in only human cortical interneurons. So in summary, humans have evolved the largest prefrontal cortex and transmodal association networks among primates, but we have also molecularly tuned conserved cortical cell types to acquire new properties and likely dramatically expanded connectivity. Thank you for your attention.